Hello everyone, thank you for attending to today's bioCADDIE external webinar. Today's speaker is Dr. Stephan Schürer. He received his PhD in synthetic organic chemistry from the Technical University of Berlin and studied chemistry at the Humboldt University and University of California, Berkeley. He's a director of drug discovery at the Center of Computational Science and associate professor in the Department of Pharmacology at the University of Miami. The research focus of the Schürer group is in systems drug discovery. The group integrates and models small molecule protein interaction, system biology omics, and chemistry data to improve translational of disease models into novel functional small molecules. If everyone could please mute your microphones unless you're asking a question. And Dr. Stephan, feel free to go ahead and get started.
Thank you, Diana, for the introduction. Welcome, everybody. I hope you can hear me. So a great pleasure to give the webinar at the bioCADDIE series today. So I'm giving this presentation as a representative of the BD2K Links Data Coordination and Integration Center. Also, I will mostly talk about what we do in the group over here. This is all an effort from 3 groups, which include Avi Marjan in New York at Mount Sinai, Mario Medvedevich in Cincinnati, and my group over here at the University of Miami in the only state that doesn't have snow. So to start, I just want to briefly mention the Links and BD2K consortia, as we are called the BD2K Links Coordination Center. So the Links Consortium has 6 data generation centers, which include Harvard Medical School with Peter Sorger, the Mount Sinai Detox S Center, and a connectivity map at the Broad with Todd Golub, another group center at the Broad Institute with Jake Jaffe, and microenvironment, a center that studies microenvironment perturbations on cell state under Joe Gray at OHSU, and the Neuralink Center with Leslie Thompson and Jason Irvine, and this is a big consortium that has several groups involved. And we are the Data Coordination Integration Center, and as I mentioned, we are also involved in the BD2K consortium, which you know very well. It has, I think, 10 centers of excellence, and then other groups like the bioCADDIE Center. And the Broad, the connectivity map is also funded to build big data analytics tools for the connectivity map in BD2K. So our center, the BD2K, the Data Coordination and Integration Center, has 4 components. Here, I will mostly talk about this integrated knowledge environment. So this is about building infrastructure to organize all the data, make all the data accessible, and make it useful. But then we have a significant internal and external data science research effort. This includes various internal projects and also several external groups, which you can find on our website, bd2klinks.org. We don't have time to go over in detail. And we also have a significant training and outreach effort. And for example, we have a Coursera course, we have summer research projects, and University of Cincinnati also has a graduate program. And then we have, of course, as a relatively large center, an administrative component, and that is charged with really coordinating within the consortium, the Links consortium, but also across the Links and BD2K consortia. So I want to remind really quick everybody about Links. So those 6 groups, they produce data and tools, and they range from transcriptional profiling, image-based profiling, epigenetic profiling, biochemical profiling, and proteomics profiling using various different technologies like the Luminex L1000 method, RNA-seq, SWOS-MS, but also targeted MS, and various other technologies, a lot of imaging data, a lot of mass spectrometry data. And the work and the main responsibility of the DCIC is to integrate this data, develop common standards, make sure this data is ultimately useful, and importantly, that it is useful in a combined way, so that the combined data will be of higher value because it can be integrated compared to all the individual pieces. So now, during the last year, we have made substantial efforts to really implement FAIR principles with the goal to make links data really fair, as you certainly know, stands for findable, accessible, interoperable, and reusable. And in particular, this includes to have globally unique and persistent identifiers, have rich metadata annotations, so it can be identified, and have via those metadata the data and the various different entities that are connected to it registered and indexed in searchable resources, for example, bioCADDIE, but also in other resources. And we have the ability to retrieve those by identifiers in an open and free way and using standardized protocols. We want to have interoperability, so formal representation of the information using formal languages, vocabularies, and qualified references, but also in informatics protocols to retrieve and access data like APIs and so on. We of course also have user-based interfaces. And then we have reusability, so we want to have clear licenses, provenance, but also, for example, very clearly defined data processing protocols, so it's clear how data was processed from, for example, raw image data all the way to some final signature. So, so I will go through a lot of the things that we do to achieve that, but what I want to do is mention the linksproject.org website, which the DCIC is maintaining and has put a lot of effort in. This is in particular effort done by Avi Marjan's group at Mount Sinai. And it has, in particular, a marketplace for all the tools. So they are organized in various different categories and include a lot of tools from the consortium. Most of them are from the DCIC, but there are also a lot of other tools from the various other centers. And it has publication, it has basically all the information, or at least a starting point to get really all the information about this entire consortium, all the groups, all the centers, publications inside the consortium, but also publications outside the consortium, and all the available tools and links how to access data. So starting with the end in mind here, during the last year, we have developed this links data portal. Here you can see a homepage of the links data portal, which is organized by various different categories by assay methods, large subject areas, for example, like proteomics, transcriptomics, image-based, and so on. Of course, the centers, various projects, biological processes. And then you can see here, for example, methods with various different assay methods. And then you can navigate all the data sets using this hierarchy, and you can go to the data sets. You can also get to all the metadata like small molecules, cells, and so on. And then it includes for all of those selected assays a brief description of how they are done, what the methodology is, and how they work. Now the data portal is organized in a catalog. So we have a catalog. I'll show this later in a little bit more detail. We see all the different data sets here, and we can filter them by various different facets. And they can also be combined. And then importantly, we have those dataset landing pages that are really a facilitator to bring all the information that is connected to one dataset together at one place. And then those dataset landing pages include, of course, general information, but also metadata. For example, here for those datasets you see, for this one dataset you see small molecules, cell lines, genes, and then you can access the data, you can download them, and you see the different data levels here, 1, 2, 3, 4. The sizes, and they can be downloaded and accessed as data packages, which I explain in a little bit. So to implement this, we were guided by this joint declaration of data citation principles, which bioCADDIE also has been closely involved developing, and they are of course important so that we have a legitimate product of research. We want to have attributions of who generated the data. of course, we want to have credible information, unique identifiers. We need to have clear access to this, and we want to have persistence. Like identifiers and metadata should be available beyond a particular vehicle to make this data accessible, like this data portal. And we want to have verifiability and interoperability, like I explained before. So the data set landing pages are really to integrate the data, the metadata, the data processing pipelines, and to provide references to the long-term repositories and the identifiers of the data sets and all the corresponding entities that are connected to it, like small molecules, cells, proteins, genes, and so on. And all of those should have unique identifiers like URIs that correspond to ontologies, DOIs on GitHub, dataset repositories, and so on, right? And then if this is implemented, so this is work in progress. We have those dataset landing pages done, but we haven't yet implemented all of those unique identifiers. We are working on this, and we have that. We will probably have that done like in a couple of months. And then once it is possible, once it is done, it will be possible to have those dataset landing pages at a place that is really persistent, and then at least in theory, if all the places that hold those unique identifiers are there, we can, all of those will come together and will be completely possible to reproduce and connect, independent for any particular tool, all of this data and all of the associated pipelining protocols and metadata. Right, so the dataset landing page in more detail is shown here. So you have a unique ID, a name, what is the center, the investigator, funding resource, a description, what is the original data source, release date, what's the processing pipeline, how it was done, and then there are now implemented like how to cite this data set. And then you have information about the assay name, description, an assay protocol, how it was done in various different categories as well. Okay, so now I want to talk about how we go about actually putting all the data together and then putting it on this data portal. So reminding ourselves again of the diverse data that is generated in links. It is our responsibility to essentially process all of those data in some way so that they can all talk to each other, so they all have the same metadata references, and moreover, we're working towards that also have references that are valid beyond links, so there's integration is possible with other projects, right? So the first thing we did is generate the metadata standards, which are of course the foundation for data integration. So this just illustrates the various different fields we have defined for cell lines, primary cells, and so on, iPSCs, small molecules, proteins, antibodies, and so on. And so those are available on the links protocol.org site, these specifications. They are also in biosharing and are accessible to the wider community over there, and we will link them further to other resources. Now, links data levels, we have made an effort to harmonize data levels across different assays. This is of course more difficult than having it just for one data set or for one type of data, like for example you have RNA-seq or CHIP data and TCGA, but we use this of course as a guidance. So we have level 1 as raw data, and then level 2 processed raw data, and level 3 conceptually normalized per sample data, so it can be used for statistical analysis, and then a signature level that can be used for connecting the perturbations, right? So in which we can now connect like information across different spaces, for example, transcriptional, proteomics, biochemical, and so on. Right, so we have made an effort. This is still not complete because it doesn't really exist for proteomics data at all, but it's in the works to harmonize this. And of course, the goal is to make it easier for user to understand what it means, what a certain data set or data file actually contains, and also of course facilitate integrative analysis. So also, as I mentioned, the data processing pipelines, there's an effort going on to validate those. So we want to capture how each data set is processed from each stage, raw data to stage 1, level 1, level 2, level 3, level 4, and so on. And then how they can be reusable and reproducible. And for example, here you see some examples at this GitHub URL that's shown here. And that's going on for various kinds of data. Right? So now, how does the data release and publication process work? So we have a submission tool of data. Data gets submitted via a submission tool, via APIs, or we get it from some websites. And then metadata and data get submitted. The metadata, of course, is guided by the metadata specifications, which have been developed by the data working group. And then what happens is all this data gets integrated into the metadata, goes into a metadata registry. I will show later how this goes, how this exactly works, and then it gets associated with all the data sets. And then we package the data up, all the data files and the metadata, put them into dataset packages, and then they get linked to the data portal. And via the data portal, we integrate together all the information about the data and all the details which are in the database, which has an API, and then can access and connect to the data which are on an FTP side. Right? So this data processing line is operationally relatively complex. So we have data and metadata. Like I mentioned, we have different mechanisms. Then we have a validation, various validation steps where we check if the information is all valid. There needs to be various steps in processing it, formatting, mapping data, generate various files, assemble packages, and then there has to be, of course, a QC process that also has several different components, and then a release where it gets posted and made accessible to the links data portal. Now here's a screenshot for the metadata submission tool, and I presented this in one of the BD2K metadata webinars. So you have here a way to submit metadata for various different categories, and they include, of course, the specifications. You can, in the tool, you can map. This is like a development URL here. It's just kind of a little bit outdated one, but you can map essentially every single field in here that you have in your file, which is in here, to the required various fields that correspond to any kind of particular metadata category. For example, here for small molecules. Now, what we are working on, because this is not a completely automated and very smooth process, we are working on an improved dataset and metadata submission tool, and we see like a prototype screenshot over here, in which you essentially, every group would be able to see all the datasets submitted and would see if they are already ready, published, or if they need to review, or if they are pending, meaning they're still being processed. Right? And the way this is going to work is by basically having a kind of a wizard in which one submits all the metadata using this tool, and then also submitting the dataset. And the dataset has various different annotations and categories, like we show. Some of them are shown here. And then this way it will be a more streamlined way of submitting all the data and associating all the data in the data files with the various different metadata categories. And now what we are doing is working with the CEDAR group at Stanford with Mark Musen to build templates and build the technology and connect the links data portal to the CEDAR infrastructure to essentially facilitate this entire submission process. So this is what we are working on right now as one of the things in BD2K. So now, once the data is submitted via a tool or via another mechanism, it needs to be registered to generate a standardized representation. So the way this happens is basically conceptually, it goes into a staging table, and then there's a standardization process, and then a standardized canonical representation is generated, and those is then mapped to the batch representation from the center, and then there's a view is created so that every single record and every data set can be associated with its specific batch record or batch reagent, but also with a standardized representation of that of all of the relevant reagents and entities, which is of course necessary for data integration. So I can give you one quick example how this works, for example, for small molecules. A small molecule would be submitted to the DCIC via those mechanisms, and would go into a staging database. So this is all, of course, chemically intelligent, right? So we understand what the chemical structure is in that. And then there is a QC process. So we identify problematic smiles. So for those that are not problematic, there are various steps to strip off salts, strip off metals, handle multiple fragments, handle various different representations of sometimes complex structures. And then we need to process the protonation states, like protonate acids, deprotonate bases, only for the purpose of having a standardized representation. And then we can standardize the structure, with respect to charges, stereochemistry, pi systems, and so on, and generate a canonical tautomeric form. It's many heterocyclic, but also many other compounds that can exist in different tautomeric forms. So we only want one, and often they get submitted in different representations, and then they get QC'd and aggregated and combined into one single structure, and then this structure is then canonicalized, and then it gets mapped to various other resources via this canonical representation, for example, PubChem and Campbell, and then from this unique representations, we generate a links identifier, and that goes into in the standardized database table, and then this goes into this metadata registration system, which is then connected to the links data portal. All right, so for example, one didn't come through here properly, but I can just show you some of those challenges, what's happening, right? So for example, one common problem is inconsistent, incorrect stereochemistry. For example, you have a structure submitted as parsenolite, right? You see here the various different either defined or missing carbon, tetrahedral carbon stereochemical representations, and you see here what's actually the correct one, but here you would have essentially via those submitted structures different IDs, right, that probably are supposed to represent this correct structure, but you're having those kind of different errors, right? You also have, of course, tautomeric forms. Here you have two different tautomers. This would be the same CAD. So there is some, of course, functionality in other resources like PubChem to consolidate them, but here there's supposed to be another structure that I will include in the slides to be submitted, in which the hydrogen is actually here, and then it gets a different CAD, right? And of course, we need to do this. We need to standardize those in order to have a correct and valid representation, which is not always present in other repositories, like, for example, PubChem. And another issue, for example, is the geometric configuration of alkenes. So for example, resveratrol is, of course, the transform, right? But there are various different representations, either an undefined, easy mixture, or just the C configuration here. And unless this is standardized, we won't be able to map this properly to other resources, right? So here is a September 2016 release with various improvements and better error handling and better standardization, and it has like 45,000 provided samples and 41,000 unique LSM IDs, right? So now, once we have done all the standardization, we can map it to different resources like PubChem or to Campbell or via Unichem to kind of about 80 other resources. And then with that, this is demonstrating this is also used by others to map links compounds to other resources using this Unichem mapping that we have provided. And then once we have done this, we can now create annotations for those compounds, right? For example, from other resources like a Campbell database or another Common Fund project, IDG, from Drug Bank, from PubChem, and then we can provide those. And many of those annotations are now also on the data portal, and they are available. And then what we can do is also use those annotations, for example, biochemical targets, and that to make predictors and then predict biochemical targets for those. And that would allow us then to better integrate, for example, transcriptional profiling data with presumed or likely biochemical targets to understand or have one way to understand mechanism of action, or at least build hypotheses about mechanism of action. And of course, there are other ways of doing this, and several of those, several methodologies are being developed in our center, meaning our entire DCIC, to do this. Right. So now, if you look at this, what annotations already exist, and many of those are already well studied. For example, links compounds, and this is a little older older analysis here, but just to illustrate the fact, so many of those are known FDA-approved drugs. Several of those are chemical probes from the Molecular Libraries program. For many of those, we have actually PDB co-crystal structures, so meaning with the links compound as a ligand in a crystal structure. And then very many of those have actually unique target bioactivity annotations. That means we know for this compound, for a particular target, what's the IC50, the KD, or the KI is. And we have this, at least at this point here, for 1,100 protein targets. So, and then with that knowledge, of course, we can, provide users with information and with enable them to build hypotheses, for example, for mechanism of actions and what those compounds are actually doing. Right? So now we can also map this to ontologies. So we have developed a drug target ontology, and we can now map those links compounds and their targets from those resources onto this tree, and then we see like that a lot of the targets are actually hitting GPCRs, like 659. Of course, many also kinases, but it's not only kinases. Links is very cancer focused, and a lot of the compounds are kinase inhibitors, but we see here clearly that there is also a lot of like GPCR focus. We also see, of course, where there are big gaps. For example, here, these are the olfactory GPCRs, and of course, ion channels, nuclear receptors, and that, right? So this is just an illustration of those biochemical annotations. Now we have done the same for cell lines as well. So for cell lines, we work with Oliver He at University of Michigan, and we take all the link cell lines, which combining the pilot program and now the main links production phase are over 1,000 cell lines, and we have been working to adding them to the cell line ontology. And then by doing this, they can be mapped to other resources that have the cell line ontology, and then it allows to integrate this information together, right? It also allows to integrate with other resources, for example, diseases or organs and tissues, then to organize the information yet better, right? So, for example, here you see a hierarchy for organs and tissues. This is, I think, a Uberon hierarchy, and here you see a hierarchy of those cell lines based on diseases, and here you see, of course, the a large focus of links is cancer, and where those various cell lines fall in. And now with those type of annotations, of course, it is relatively straightforward to map links data, for example, to patient data with certain diseases, so links model system data from cell lines to, for example, TCGA data for certain patients with certain diseases, right? Because they share the same annotations, right? So now again, this is done all put together into the links data portal, and I have shown you the homepage here already just to recall this here. And then you can see now the various different data sets, various different groups, assays, the methods, what are the different categories. A more recent version also have all the data set links directly here. And you see that the various different like facets, assay, process, various projects, is more information down here, the method. And then we have the data set landing pages with the information that I showed you, and detailed mappings to the small molecules, cell lines, genes, proteins, and so on, whatever applies to the various different categories, and the downloads, like I mentioned. So we also have built links for molecule app and small molecule landing pages. So here you see like a similar arrangement, but now we're talking about all the small molecules, for example, and we have a similar app for link cells. Cell lines and other cells. And for example, you see here now what are approved drugs, and we have now recently included a lot more clinical annotations, for example, like what clinical phase it is, clinical phase 1, 2, or 3, and various kinds of other pharmacological annotations, which you can see if you go to the links data portal, which is linksportal.ccs.miami.edu. And here we see, of course, a structural representation, various different links to the centers. And then we see all the different batch representations, like I mentioned. So which particular provider and which particular batch representation and ID was used from the various groups for those data sets, and we have all the different data sets linked for a compound. Here, sorafenib, of course, is a approved kinase inhibitor drug, is profiled extensively in various different links data sets. Right, so and now there are also APIs on the links data portal, and then via these APIs and standardized representations of all of the metadata that are connected to this data sets. Now all the links data sets are also indexed in the bioportal in bioCADDIE, and we have worked together to basically make links one of the first data sets that are indexed in the bioCADDIE data mat system. And as you can see, you can query it and then get those various different data sets and the link back to the links data portal. Now, so I'm going faster than I'm expecting here, but maybe we have more time for questions if you have any. So now, once we have done all of this data processing, what I want to mention, and that is important, of course, we now be doing this all for the purpose of being able to enable users to properly analyze links data, right? And one of the integrated analysis tools, environments for links data is iLinks, which is a tool developed by Mario Medvedevich in Cincinnati. And for example, here you can query links data from various different perspectives. For example, genes, what are my genes, proteins doing in links data? What are the different data sets which are linked to the data portal, right? What are they doing? And then can I analyze those signatures? So can I find like concordant, discordant signatures to certain diseases, construct consensus signatures, and so on. So this is a very powerful tool that allows to really analyze links data interactively and in quite a lot of detail. Then, yes? Oh, okay.
If everyone could please mute their microphones unless you're asking a question. Thank you.
So, yeah, I think we will have plenty of time for questions, and I'm happy to elaborate further on any of those tools if I can. So now this links this iLinks system, this iLinks analysis environment now allows various different visualizations like clustering, PCA views, and pathway analysis, volcano plots, and all of those different types of ways of looking at it, and also connects and integrates with other tools that are developed in the links consortium. For example, the Enricher tool, which is probably the best gene set enrichment analysis tool that is out there, and that's been developed in Avi's group for a long time, Avi Mayan's group at Mount Sinai. Or, for example, also a signature search tool. It's an L1000 CDS square, which allows you to basically put in a gene signature, a gene transcription signature, and essentially find concordant or discordant, meaning reversing or mimicking perturbations among all the links data in a very easy, user-friendly way, right? So now this is just those types of tools that we have now. We are building them, integrating them together so they can be workflows. For example, here you see a workflow from the links data portal, an RNA-seq data set going to iLinks, then allows you to analyze differentially and identify differentially expressed gene signatures, and you can go to Enricher, see how they get enriched, look at different pathways, look for mimicking or reversing small molecule signatures using the L1000 CDS square tool, for example. And connect to various other kinds of analysis tools. Just as one example, right? So the links TCGA data, links RPPA data, and so on, right? So not links TCGA, but TCGA and RNA-seq data, and links RPPA data, which is also a data set, a proteomics data sets that are being generated in the consortium, and they can be connected. Now this is just one tool of many, and I touched on two other ones. So the links project, and again in particular, our center has developed a large suite of tools. And if you go to the links.polytech.org website and links tools, then you see basically a lot of tools, and they're organized by various different categories. And again, they have been developed by really all the groups, all the data generating, and of course our data integration center. And it allows us to their range and functionality from querying data, integrating data, visualizing data, interactively analyzing data, or computing on the data. So now what I want to mention though as well, and what we are working on at the moment is it's still challenging despite those tools, and this is of course reflecting that there's a lot of different apps. There are specialized apps to do something with any of those data sets. It's of course one of our eventual goals is to really integrate all, or at least various different types of data sets and links together. So we want to have a way to efficiently organize and store all links data and all the link signatures. And for this, we are developing this link signature store. And for example, this is already now at the point where it allows us to query and to retrieve any link signature, any data point, or any combination. We can already, flexibly and fast compute with link signatures. And importantly, we want to integrate across different link spaces, and meaning that, like, for example, transcriptional space, proteomic space, biochemical space, or chemical structure space, right? So we can find signatures that correspond to certain perturbations, or certain compound perturbations or genetic perturbations, or share different disease models, for example, cell lines, and so on, and then have all of those signatures in a representation that allows to directly compute on them and to query them in a flexible way. And to do this, we have built a big data analytics system. I won't go into detail, but it's basically a Hadoop Spark-based system to organize all the information. And essentially, that now enables us to have an end-to-end data processing pipeline in which we have start from the linked data portal packages and the standardized metadata, and we then process the data, aggregate the information, load them into the signature store, and then on top of this have APIs and RPackage, Shiny apps, and so on to really allow others to interoperate with the data, but also to build their own tools with that data. And then, of course, what I showed you before is how do we get those linked data packages, get all the standardized metadata, perform all the standardization, all of this. So at the end, we really want to have, and we will have that complete end-to-end pipeline from data submission via different mechanisms to registration, standardization, data set packaging, to then processing all the data in a way that we can actually operate on those final high-level signatures and integrate them together. The current status of the signature store is we have about 2 billion data points in the 70,000 entities. It includes L1000, P100, GCP, and various other data, all the metadata, chemical structures, and there are a couple of available applications already. And we are now in the process of adding a huge amount more of L1000 transcription response data, and we will also add the known, like I mentioned to you, the known biochemical targets, and also predictions in there, so it would allow to really integrate, for example, the transcription responses with biochemical targets in a better way. All right, so with this, I'm already at the end, and I just want to thank everybody who contributed to this, without mentioning all the individual names. But I want to say that it's really, again, really a team effort across the entire DCIC, which includes Mount Sinai, Cincinnati, University of Miami, and if you want to see all the people, they're on the bd2carelinks.org site on the team. I thank you for listening, and if you have questions, I will do my best to answer them. Thank you.
Stephan, this is Ron Margolis. Can you hear me?
Yes, I can hear you well, yeah.
Good. This is fantastic. I've been familiar with links for some time, and in particular as it works with bioCADDIE. My question is sort of a general one. Have you been able to gauge the relative community utilization of the links portal and the tools and data sets that are to be found there?
Yep.
And in particular, is it possible to, at this early stage, get a feel for the value added to the broader community from this.
Yeah, so regarding the first question, so we we have, of course, Google Analytics on those tools, and I don't have them right in front of me, but some of the resources, for example, the web page, of course, is really widely accessed. I think it has like 20,000 users, unique users, and twice the number of sessions or something like that in the last quarter. Now the Links data portal is still a little bit newer, so we don't have that many users, but we're capturing users and we're also capturing downloads and all of that. And what we are doing now is we are really kind of considering to rather than just going for mass, also going for depth, and seeing if we can put together like something like more like a Links user community. And if we, for example, could convince people to register, give us some more information, and then kind of make like a more in-depth community that it will be a little bit smaller and not like tens of thousands, but about which we know a lot more, right? And then we can work with them closer to gauge what you said, like what's the value add and what the wider community really wants to do, right? So my personal opinion is that I think it is of course important, I think, to have all of the data sets and data packages and everything really well organized and have all of the metadata and everything connected and have all of this available and have provenance, right, and all of that. But my best guess would be that if most of the users will be interested more in the final results, right, and they want to query more like based on cell lines, diseases, small molecules, particular genes or proteins. And there are already many tools that allow to do that. And of course, the goal of the signature story, as showed you, is to provide that to link all of those final endpoints to all the data sets, right? So for example, then you can query something, right? You can do some analysis, and it would be immediately clear because we have the provenance of all of that. What are the data sets that contain this information, and then how should you be citing that, right? As my data, that I used comes from those and those data sets, right? And that's kind of what we are working on now to kind of almost like close the loop, right, to provide the maximum value for the community that really has not just the data sets, but also every individual, at least the high-level data points, right? Every gene perturbation, protein level, biochemical target, and whatever information we have, IC50 or percent control, and so on, right? Lots of data and links. I mean, I hope that's kind of like a satisfactory answer.
Oh, yes, it is. This is a problem I've seen in other contexts as well. If you put it out there, do people actually use it? And how can you assess how it's being used? And thus, how can you evaluate the value of the materials and capabilities that you've put forward?
So I think one other thing I forgot to add. So we do track, and again, with all of this I'm talking about the DCIC. We do track the usage, of course, but also publications. There's, of course, is also a certain manual effort to capture those publications. And I think to say what's the value add is probably something that will be much clearer in a quite similar way to the MLP program, which you know very well, right? Probably like a few years later, right? Because all the papers that come out, all the utilization really. Also now we have I think there are a good number of community publications. It's quite a reasonable assumption that there are probably 100 or more projects that use links data ongoing right now that will probably lead to a much larger number of additional publications. And then this type of community engagement with the data will only increase as we move forward.
Yeah, I agree.
Stephan, this is Anu from bioCADDIE. Great talk. I have a quick question regarding the tools that you mentioned. So do you have linkages between what data sets could utilize which of the tools that you actually have?
Well, that's a very good question. Actually, this is a project. I didn't have time to mention this, but this is actually something that is that we are implementing right now. So there will be on the data on the links data portal site, there will be tools that are applicable to those different to the particular data set in the data set landing page. But then another project I should mention is that Avi's group is working on is having data set to tools, having and actually data sets to analyses, right? So going one step beyond, showing what kind of analyses do exist for what kind of data sets, and then what tools have been used to do those, right? So there will be pre-canned analyses of the various data sets, and then of course the benefit of that would be that now you can directly connect them, right? So it becomes a lot easier, even though they are pre-made. For a large chunk of the users, perhaps the majority, this may be what they exactly want, right? So we will have both various analysis mapped to data sets that have already been done, and then also what tools are applicable to the various different data sets, and that is already there to a certain extent. If you go to the tools, you will see get a sense of that as well, what data sets are behind those various different tools.
So following up on that, I wonder if this is part of the metadata that's provided to us from links for indexing, because that's something that would be useful to display.
So I think we could do that via the data portal once we open that up and have all of the tools mapped for each data set. And then you could imagine there is a BD2K project, right, that catalogs all the different tools, and then with that catalog of all the different tools and the catalog of the different data sets, right, you could provide that connection, and bioCADDIE may be a good a good tool to actually make that connection, and you could certainly get it from us from links.
All right, thank you.
Are there any more questions? Okay. Well, thank you so much, Dr. Stephan, for your presentation. Thank you, everyone, for attending. I will see everyone for the next external webinar, which is in February the 9th. Thank you, everyone.
Bye.
Thank you, and feel free to email with questions. Okay, bye-bye.
Dr. Stephan, goodbye.